So it is my distinct pleasure to introduce Ava Amini. I believe she actually helped Kevin start this seminar series a couple years back. So we're really excited to have her here and talking about some work that she's done at Microsoft. Ava is a principal researcher at Microsoft Research in Cambridge, Massachusetts. Her research focuses on developing new AI methods to better understand and design biology with the ultimate aim of realizing precision biomedicines that improve human health. In addition to research, Ava is passionate about AI education and outreach, and she is a lead organizer and instructor for MIT Introduction to Deep Learning and an in-person and global course on the fundamentals of deep learning. So Ava, we're super excited to welcome you today and whenever you're ready.
Thank you so much, Meg and Leo, for the invitation. And it's my pleasure to be here. Indeed, Kevin, myself, and a few others a couple years back had the idea to start this ML for Protein Engineering seminar series, and it's amazing to see just how much the community has grown over the past years. So today I'm going to present one of our very recent works that has actually been a really fun joint collaboration, bridging computation and experiment between our group at MSR and Sankita Bhatia's lab at MIT, which is where I completed my PhD. So it's been a fun way to kind of bridge the worlds that I've stepped from the experimental world to the computational world. So just to start, I want to contextualize a little bit about the scope of this work in terms of our broader research agenda in biomedical machine learning at Microsoft. And fundamentally, we are a science-driven team that is focusing on the development of new generative AI systems and methods to enable biological design and discovery. And most relevant to this seminar series and to our talk today is the efforts that we have taken at the molecular scale in protein engineering and design. And so you may be familiar with some of these works that we've focused on over the past couple of years, including the development of foundational generative models for protein sequence design or structure design, as well as sequence-to-function data sets and annotation methods. But today, I want to do something a little bit different. Rather than focusing on the development of these foundational approaches, talk about some work that we've done recently in bringing these principles to targeted biological applications, which have been of great interest to us recently. And in particular, we're quite curious about enzymes, not only from the perspective of enzyme design, but also in designing new substrates and sensors that can measure the activity of these enzymes in biological contexts. And so today's talk, we're going to dive deep into one such class of enzymes, which are proteases. And proteases are this incredible, highly multifunctional class of enzymes that play critical roles in both health and disease. And fundamentally, the catalytic activity of proteases is to cleave peptide bonds. So to cut proteins, cut short peptides, and this triggers signaling, is involved in cell regulation and several processes in disease. Now, in the human proteome, there are 566 unique proteases, which fall into 5 broad catalytic classes based on how they function and their catalytic mechanism. And so today we're going to talk specifically about metalloproteases in specific, and you'll see that become important later on in the talk. Now, because of the criticality of proteases in maintaining biological function, what that means is that across several diseases in both infectious disease and in cancers, protease activity can become highly dysregulated and play actually key functional roles in the progression of these diseases. And so perhaps the most notable example of this canonically is in the context of cancer. It's been known for decades that tumor cells and cells of the tumor microenvironment can actually produce and secrete proteases that activity is aberrantly regulated. And as a result of this, because the activity of these proteases is to break down other proteins and cleave peptide bonds, proteases can actually play key roles in breaking down the tissue of the surrounding tumor microenvironment, which helps cancer cells spread, tumors to grow, metastasize, and actually facilitate the invasion of the cancer. And so not only has this been particularly interesting from a biological point of view, but in recent years, people have sought to leverage this dysregulation of protease activity that's specific to the tumor as a diagnostic or therapeutic target. And so one very promising approach for thinking about this is to actually leverage this dysregulated protease activity as a trigger to selectively activate a therapeutic or a diagnostic such that it acts specifically within the tumor microenvironment to attack the cancer and minimize the off-target effects. And so the general engineering principle behind these approaches is that you have some sort of agent whose activity is blocked by another peptide or another functional group. And you can link this blocker to that diagnostic or therapeutic agent via a peptide that ideally can be selectively and efficiently cleaved by those proteases that are specifically activated in the tumor microenvironment. And so in principle, right, if you have this triggered activity, you can now leverage this as a signal amplification or triggering mechanism to now activate your agent in the context of disease. And so one fundamental engineering challenge here is to design these actual linkers that can be efficiently and selectively cleaved by the target proteases to optimize the activity of these agents. Now, this gets really challenging really fast, right? Because proteases recognize peptide bonds, that means that their activity can be highly diverse, highly complex, and often very promiscuous. And so to dive a little bit deeper into how these cutting events happen, right, remember that proteases are just enzymes. And so we can think about peptide recognition from a traditional sort of lock and key mechanism. And each protease within its active site will have different pockets that can preferentially recognize different amino acid building blocks. And if there's a right match, the protease will recognize that peptide substrate and cleave it in an active way. Now, a first challenge here is to think about, okay, what are the possible substrates that can actually meet this criteria of a match to the active site? While this example shows one possibility, there can actually be multiple promiscuous substrates that still fit within the catalytic site of a protease. Even further, if we think about just a short peptide region of, let's say, 8 positions of amino acids, the complexity over this design space quickly scales combinatorially. Because if we consider, our 20 natural amino acid building blocks, we can quickly reach over billions of unique sequences that could be possibilities for just a short 8-mer peptide. And so from an engineering standpoint, there's this first question of how can we actually screen and design, narrow down this search space of possible peptides to design these cleavable linkers. A second challenge comes when we consider the fact that usually it's not just one protease that's dysregulated, but multiple proteases. And perhaps we want to design for recognition by multiple of these proteases, or rather optimize for specificity to one protease. And so this presents a second engineering challenge from a design point of view of how do we ensure both efficiency of cleavage and selectivity. And so traditionally in biochemistry and in sort of chemical biology approaches to design substrates for proteases, the most dominant approach to substrate nomination has been basically manual curation and nomination based on literature review and some small databases of protease substrate activity measurements. However, this quickly becomes limited not only from a throughput point of view, but also in terms of the established evidence about the specificity determinants of both the proteases and the substrates that they recognize. So there are clear limitations with this kind of hand-curated expert-driven nomination approach. Conversely, in recent years, we have seen the emergence of high-throughput experimental methods like display-based strategies, for example, yeast display, phage display, mRNA display, that effectively amount to trying to screen thousands or tens of thousands of peptides in vitro and leveraging automated experimental setups to measure the activity of these substrates against target enzymes. The challenge here is that oftentimes these display-based strategies are very specialized, require a lot of prior knowledge and experimental expertise to actually deploy. And so for a general practitioner who may not be, proficient in something like yeast display, this can also be limiting. And so looking at this design problem, we were motivated by the emergent work that has been going on over the past years in protein language modeling, and considering taking inspiration from these approaches to now not think about protein design, but rather peptide design in the specific context of protease substrate engineering. And so our motivation here was to develop a deep learning pipeline that would learn over the amino acid sequences of these peptide-based substrates to not only enable generative peptide design, but also rapid in silico screening of these candidate substrates for their predicted activity relative to target proteases. And so to that end, we reasoned that we could tackle this through a kind of joint approach, where we not only developed a generative model that would learn from protease substrate data to now be able to propose tens of thousands or hundreds of thousands of individual candidate sequences, but also combining this with a predictive model that would enable rapid in silico virtual screening of these candidate designs to now narrow in and nominate sets of these substrates for experimental validation. And so that's exactly the approach that we took in this recent work, CleaveNet, which was, again, a really inspired, bridged effort between computation and experiment, bridging our group at MSR with Sangeeta's lab at MIT. And so the overall principle is exactly as I outlined on that prior slide. Our goal is to be able to go from a target protease of interest and leverage a generative model that can now produce tens of thousands of de novo designed candidate peptide substrates, and now be able to couple this rapidly with a predictive model to screen and down select these substrates for particular applications and experimental validation. And so, as you can probably appreciate from this layout, the CleaveNet pipeline consists of two components. The first is a predictive model where, given an input amino acid sequence, we can generate continuous cleavage scores across a range of protease enzymes of interest. And we couple this with a generative model from which we can either sample unconditionally to generate new sequences that are going to be in distribution of this possible design space. And ultimately, what we'd really like to be able to do is to do guided or conditional generation of candidate substrates based on a desired protease cleavage profile. And you can envision that this becomes really powerful because we can now specify a set of constraints according to the activity profiles we want to optimize for, and hopefully be able to produce new substrates that will match that desired activity profile. And so today I'll go through this kind of end-to-end pipeline, both from a modeling perspective in terms of how we developed and validated the predictor and generator models in silico, but also an experimental perspective in terms of how we have actually brought the generations from CleaveNet to the wet lab and validated them and deployed them through large-scale in vitro experimental screens. Okay, so that sets the stage. And so to get into it, we'll first talk a little bit about the predictor model. To do so, we first have to consider, what data that we're actually going to train both the predictor and the generator on, and ultimately validate our methodology on. And so to do a first pass at this, we decided to focus on a particular class of proteases that I introduced earlier across these 5 distinct catalytic classes. And that is the class of metalloproteinases, or MMPs. And in particular, we were motivated by this because we were able to find a fairly large scale, at least in the protease community, large scale data set of displayed short synthetic peptides screened against a set of 18 distinct matrix metalloproteinases. And so in particular, this is a data set that was actually kind of buried in the biochemistry literature from 2015 of this library of approximately 18,000 synthetic 10-mer peptides that were displayed and screened through a mRNA display approach. And the particularities of how this assay is set up are not especially important. What is important to keep in mind is that from this data set, we are able to actually get a continuous metric of cleavage efficiency for each of these 18,500 synthetic peptides against each of these 18 MMPs. And so we were very excited to find this data set and decided to leverage this as our base training set and also hold out a subset of this data for validation and testing. We also wanted to test the ability of CleaveNet to generalize to completely distinct experimental setups and biochemical assays. And so to that end, we leveraged a completely independent test set that was brought together in the Bhatia lab, which consists of a totally distinct assay setup. And here, the substrates are not displayed by mRNA display. They're screened individually, where we have a fluorophore tag on the substrate such that cleavage by a particular enzyme will trigger fluorescence, allowing us to generate these cleavage profiles. And importantly here, the length of these peptides differs, and we have 71 of these screened independently against these 7 distinct MMPs. And so with this in hand, our first task was to actually train the predictive model on that mRNA display training set. And the task that we set up was fundamentally a multi-output regression, where our task is, given a particular amino acid sequence S, we are training the model to predict a continuous cleavage score Z hat for each of these 18 MMPs M. And again, we leverage our 2 independent test sets for evaluation. And we did a pretty thorough hyperparameter optimization over the predictor model, and also looked at both a LSTM recurrent-based architecture as well as a small transformer-based architecture. And ultimately, we went forward with a very small transformer, only about 60,000 parameters. And this is sort of the base architecture for both the CleaveNet predictor and the generator. So a quick snapshot of how the predictor performs. I won't spend too much time on these results, but we first asked, okay, can we actually predict these continuous z-scores with high fidelity? And it turns out that we can. This is a very tractable regression problem. So what you're seeing here is the scatter plot showing the correlation of the true z-scores for one such MMP, MMP13, against the predicted z-scores. And we see we have quite good concordance across the test split of this mRNA display dataset. We can also turn these continuous cleavage predictions into sort of a thresholded binary label of cleaved versus not cleaved. And here we can see across both the mRNA display and independent fluorescence test sets, we get good predictive robustness when deploying the CleaveNet predictor model. And so here I'm showing just an example of results for one MMP, MMP13. Of course, we evaluated across the spectrum of distinct MMPs that we tested on, and all those results are included in the paper. So really our goal was not to set out to develop, some highly sophisticated predictor model, but rather to deploy the predictor within this pipeline of going from generation to nomination of a candidate set, meeting a targeted, cleavage profile for proteases of interest. And so the bulk of our efforts and the bulk of this talk are going to be spent discussing our approach to actually design this generator and validate it in vitro experimentally. Now, as I said at the beginning, ideally we want to be able to train a model to learn and sample in two sorts of ways. One is what we think of as unconditionally, meaning that we want to train the generator model to learn the distribution of amino acid profiles across these peptide substrates, such that at inference time, we can sample from it to generate new candidate substrates. And so again, for the unconditional generator, we again turn to this mRNA display dataset. And to train this model generatively, we turn to what has now become sort of the classic language modeling objective in training generative autoregressive language models. And so given that this is, an interdisciplinary audience and we're bridging protein engineering and ML, I just wanted to do a quick refresh on what this objective of next token prediction and autoregressive generation actually means. And so looking at natural language as the inspiration of training these generative models, the task is quite straightforward. We want to develop these generative models that learn an underlying probability distribution of language patterns in text data by learning to predict the most likely next token in a sequence of tokens. So what that means is, given an input token, for example, the model predicts the next word, and we feed this in autoregressively to basically grow the input and sequentially predict the next word. Now, as you probably very well know, this has translated to immense promise in the realm of protein design and protein generation, where recent protein language models have taken this principle to now achieve the generative design of new proteins by learning over amino acid sequence space. So now, in this context, if we model an individual amino acid as a dot, our task is analogous to what we showed before. We're trying to predict the next amino acid in a sequence, and we can do this in an autoregressive manner to actually produce new sequences. So we took this principle, right, which has now been, quite established in the realm of protein language modeling, to actually build out the CleaveNet generator, which again is a small transformer trained autoregressively to generate these new protease substrates. When we did this, we first wanted to consider, okay, right, this is a relatively small problem. This is not a foundational, general purpose protein foundation model. We want to focus our evaluations on the specific types of tasks and metrics and criteria that we care about from this problem of protease substrate design. And so that really guided us in thinking about, first, whether or not these sampled substrates that we can produce from the CleaveNet generator exhibited the biophysical properties that we would expect from protease, peptide-based protease substrates, whether they were novel and diverse, but also whether they recapitulated known cleavage motifs that we know to be very important in driving the recognition of these peptide substrates by MMP proteases. And finally, we asked whether we could actually put this together with the CleaveNet predictor to come up with some in silico surrogates of whether these generations were likely to be cleaved by MMPs, both in silico and in vitro. So walking through these evaluations, again, this is of this unconditional model where we've just trained it, in this general purpose way on this data set, and we're sampling from it unconditionally at inference time. So the first thing that we were encouraged to see was what we saw when we inspected the position-wise distribution of amino acids across our generated sequences. And so in these seq logo plots that you'll see throughout the rest of the talk, what we're visualizing is the amino acid distribution in a position-wise manner across these 10 mer peptide substrates. And here we're measuring the KL divergence relative to natural sequences from the mRNA display test set. And perhaps you may not be used to looking at protease and MMP substrates in specific, but there are some canonical examples, for example, the proline at P4, the leucine at P1 prime, that come up in these generations, which we were very encouraged to see. I'll also note that this nomenclature indicates that P1 through P5 are the amino acids preceding the cut site, the actual cleavage site for a particular peptide, and P1 prime to P5 prime are the subsequent positions. Furthermore, we looked at scoring these generations for their predicted biophysical properties, including charge and hydrophobicity. And again, we were encouraged to see that, generally speaking, we are recapitulating the distribution of these biophysical properties across our generated sequences relative to the true natural substrates, or the substrates from the mRNA display set. Importantly, you may ask, okay, well, is this generative model actually learning something beyond what you may be able to capture by considering these amino acid distributions in a position-wise manner, right? The seq logo only shows you the distribution completely independently in a position-wise way. And so to assess this, we set up a baseline where we sample from that exact distribution from the mRNA display dataset in a site-independent and position-wise manner. And so you'll see throughout the talk that these baseline generated sequences are denoted in yellow. And so we asked, okay, are we actually capturing some interposition dependencies from our generative model that we may be missing by just doing this site-independent sampling baseline? And so to assess that, we did sort of a saturation analysis where we considered across the space of possible k-mers, of short motifs in these sequences, how quickly do we saturate the distribution of possibilities, if we do, across the number of generations that are produced? And so this is a visualization of that behavior here, where you can appreciate that at short k-mer lengths, so for a motif or a subsequence of length 3, all three of these methods, whether they are CleaveNet generations, the true mRNA display sequences, or this baseline, quickly saturate and capture the complete space of these possible 3-mer subsequences. But as the length of these k-mers grows, the behavior of this site-independent sampling approaches linear behavior, suggesting that this is closer to a random distribution of these k-mers at these longer lengths, whereas with our CleaveNet generations, we are consistently capturing the behavior of the true substrates from the mRNA display dataset. And so this was encouraging and suggesting that we're starting to pick up on these intersite dependencies beyond the site-independent baseline. Furthermore, we can inspect this a bit further in actually generating these substrates and then scoring them using our CleaveNet predictor and analyzing the sequence composition and how that composition differs across substrates that score highly for distinct MMPs in this data set. And so here we considered two things. First, what those distributions looked position-wise, again, looking at the top scores for a particular MMP of interest. And secondly, again, from this question of whether or not our generations were capturing these critical inter-amino acid relationships. And so first, looking in a position-wise manner, what an analysis we did that gets biochemically quite interesting is now looking across these distinct subclasses of MMPs and particular enzymes in particular, looking at what the high-scoring generations for those MMPs look like versus the high-scoring true substrates that were actually cleaved most efficiently by a particular subclass or individual MMP of interest. And even qualitatively looking here, you can first appreciate that there are distinct sequence compositions and motifs that come up across these protease subclasses. And furthermore, that the patterns that appear in our generated sequences are matching those of the true high-scoring substrates, again, with distinction and separation across these protease subclasses. And finally, considering now from an inter-amino acid dependency point of view, we again performed an analysis of these subsequences, now looking at these three mer motifs. And here I think this example illustrates it really well, that even with unconditional sampling, our CleaveNet model is able to pick up on these canonical determinants of MMP efficiency and specificity, whereas samples from the site-independent baseline are completely missing these core components that are driven by these key inter-amino acid relationships. And so this was quite encouraging to us from an in silico perspective that, we're not trivially learning to just sample from the distribution in a position-independent way, but rather that CleaveNet is actually capturing these subtle and nuanced site dependencies. So now comes the fun part, actually bringing these substrates to the lab and screening them experimentally in vitro. And here we wanted to kind of focus on a particular biological test case and a particular protease of interest to highlight the power of the CleaveNet approach to do this really focused sort of design. And so first we said, okay, can we nominate substrates that would be efficiently cleaved by one particular MMP, MMP13? And here we now want to bring our joint pipeline to bear on this task, where we set up a pipeline where we sample from the CleaveNet generator unconditionally, leverage the predictor model to score them for their likely cleavage efficiencies by MMP13, rank order them, and filter down to a set of novel and orthogonal candidate substrates for in vitro screening. And here our experimental setup is we can synthesize these synthetic substrates with a fluorophore on one end and a quencher on the other end, such that, again, cleavage by MMP13 will trigger fluorescence, and we can read this out via a fluorescence assay. So in particular, we use this approach to nominate 24 distinct sequences that we assessed would be highly likely to be efficiently cleaved by MMP13, and looked at a number of baselines. Thinking again about our site-independent sampling approach, either alone, as I showed before, or by actually leveraging the predictor model to score and rank order those that were predicted to have high cleavage efficiencies by MMP13. So this is a much harder and stricter baseline than just that site-independent sampling. And we also included some positive controls of highly cleaved substrates from the original data set, as well as negative controls of uncleaved substrates. So now getting into some of those in vitro results, the first thing that we were very encouraged to see was that all of our CleaveNet generations were cleaved efficiently in vitro by MMP13. And this was true for both the unconditional samples, which we then filtered down based on predicted efficiency, but also perhaps unsurprisingly, in actually leveraging the site-independent baseline plus the predictor model to rank and hand nominate some high-scoring substrates for cleavage efficiency as well. And further confirmation of some of the sequence level analyses that we were looking at previously came through when we actually looked at the sequence compositions of the highly cleaved substrates versus those that were not cleaved. And you can appreciate that, again, there's a very distinct separation of these sequence compositions that is driving this cleavage that we observe in vitro. What is interesting now is, okay, well, here we were only selecting based on efficiency, nothing about selectivity or specificity for MMP13. And if we now start to think about, okay, we have this evidence of MMP13 cleavage in vitro, what does this mean potentially for other MMPs, keeping in mind that these proteases can be highly, highly promiscuous? And so here we did an analysis where we leveraged the predictor model to in silico score these same efficient substrates that were cleaved well by MMP13 in vitro for their likely cleavage by other MMPs in silico. And what you can appreciate is, right, there's no single bar that is shooting up and other bars going down, right? There is a clear indication of predicted promiscuity of these substrates across this range of MMPs. Now, again, this is very consistent with the biochemistry and what we know about these proteases and how they have these broad and promiscuous and overlapping cleavage preferences. And so this really motivated the principle of trying to guide the design of substrates that would not only be efficient, but also selective. And so this was the final thing that we considered, and really the ultimate goal of the project overall, to be able to condition generation based on a target cleavage profile of interest and generate new substrates given this additional input information. And again, we were very motivated by the use case and application for a tool like this in our evaluations and in our choice of modeling approach that we undertook. And so to achieve, this conditioning principle, there are several approaches that one may employ, but the particular one that we focused on was really motivated by the unique nature of this mRNA display data that we had on hand to train the model. And so the approach we took here was this idea of conditioning tags, where now, again, if we go back to our natural language example, you can think of, our standard next token prediction task, but perhaps we want to supply some additional context or information to the model about the particular type of generation that it should produce and therefore constrain the sample generations in that way. And so the principle here is to leverage these conditioning tags as inputs, which can be class labels, continuous labels, different forms of auxiliary labels given a now sequence input in concert. And so coming now to our setting of substrate design, the beauty of our mRNA display data and of this problem at hand is that we can leverage this conditioning tag principle to now supply a vector of these cleavage scores or cleavage preferences across our proteases of interest as an additional input to the model to now constrain the generation in this way. And this is exactly what we did, leveraging again this mRNA display dataset, where now instead of pure unconditional training or generation, at training time and at inference time, we can supply these conditioning tags that are vectors of cleavage scores across the 18 MMPs from the dataset and use that as a means to guide generation. And so in practice, the final CleaveNet generator is trained half of the time unconditionally without a conditioning tag as input, and the other half of the time conditionally using a vector of 18 cleavage Z-scores as a input conditioning tag. And so what this enables is a really powerful capability at inference, right, where now we can guide generation towards a target cleavage profile. And so we deployed this principle, again, using MMP13 as a enzyme test case, but now saying, okay, can we achieve the targeted design of substrates that will actually be highly selective for MMP13? And to do this, we computed a metric of selectivity, leveraging the data that we had, where we defined the selectivity score of a substrate, S for a protease M, as the cleavage of this sequence S by M relative to the average cleavage across all other MMPs. And again, we want to consider the performance of our CleaveNet conditional generator relative to a series of baseline approaches, where now we up the ante a little bit further and consider not sampling site independently from the total distribution, but rather looking at just the top 24 most selective MMP13 substrates and performing the same site independent sampling, and also comparing to generations that were produced unconditionally from CleaveNet. And so first, we tested things out in silico, where now we are using our predictor to score these generations and transform those cleavage predictions into a selectivity score. And what you can immediately appreciate on the far right in dark green here is that the conditional generations achieve the highest predicted selectivity scores. Again, this is only in silico, relative to both the unconditional generations and the total distribution of mRNA display peptides. And that's exemplified further, where now, again, if we zoom in on the sequence level composition, we see now a new motif appear where the generations that we're producing for MMP13 selectivity seem to be dominated by the presence of this arginine at the P2 position, which is quite different and distinct from the total distribution of MMP sequences we started with. Another line of evidence was to consider, okay, well, are these generations actually novel, right? We seem to be picking up on some interesting motifs and site dependencies, but are these generations new? And here we again looked at an analysis of k-mer subsequences, and we found that actually, if we think about k-mers of increasing length, the generations occupy a completely distinct set of k-mer motifs that appear in these generations relative to the sequences that we learned from. And so this was quite encouraging to us, giving us some evidence that, we could generate sequences that were not only likely to be selective, but also ideally novel. So now we said, okay, now is the final test. Let's scale this up. Let's put all these pieces together to see if our approach actually yields substrates that are going to be selectively cleaved by MMP13 in vitro. And so we significantly scaled up our in vitro experimental screen, where we compiled a list of 95 distinct sequences spanning some of these MMP13 efficient generations from the unconditional plus ranking approach, as well as some of these conditionally generated sequences that were hopefully optimized for MMP13 selectivity. And in our screen, to assess both efficiency and selectivity of these substrates against a range of MMPs, we did a pairwise screen where for each of these 95 substrates, we measured its cleavage against each of 12 distinct MMP proteases. So this gives us a large matrix of these cleavage profiles spanning substrates by proteases. So to give you first a kind of a high-level snapshot of what that data looks like, we're going to display it on a heat map where blue dark entries indicate substrates that were not cleaved, and red indicates substrates that were highly cleaved in vitro in this screen. So the first, sanity check and encouraging thing we saw was if we looked at our negative controls, substrates that we expect to not be cleaved by any MMPs, across the board, they're not cleaved by MMPs. Now, if we look at the substrates that we nominated for efficiency, we can see in general that for those generated by CleaveNet, denoted in green, we see a good spectrum of cleavage across these MMPs, but not necessarily selectivity for MMP13 here in the first column. In contrast, when we look at our substrates that are designed for selectivity, we see lower cleavage overall across the spectrum of the MMPs, but encouragingly, just from this heat map, we can already begin to appreciate that there is potentially selectivity for MMP13. Now we can dive into these results further and display these individual cleavage scores for MMP13 measured in vitro in what we call a selectivity versus efficiency plot, where on the x-axis you have cleavage efficiency by MMP13, and on the y-axis is the selectivity score for MMP13 relative to all these other MMP proteases. And so we use the empirical determination to kind of devise these thresholds for efficiency and selectivity, separating this grade into 4 quadrants. And to start, when we look at this bottom left quadrant of substrates that are neither efficiently cleaved nor selectively cleaved, it tends to be dominated by either sequences from the baselines or from some sequences from the mRNA display dataset. In contrast, when we look at the bottom right quadrant, where now we have high efficiency but low selectivity, we were encouraged to find that our CleaveNet generations, both selected for efficiency, whether through unconditional generation and then scoring, or through site-independent baseline and scoring, appear in this bottom right quadrant. We were also finding a very high concentration of substrates from the conditionally guided model in the top left, where we have high selectivity but poor efficiency. But most encouragingly is what we observe in this top right quadrant, where now we have a handful of substrates that are high in selectivity and high in efficiency. And so our final question was to see what we could observe in terms of what these sequences were capturing if we were to now zoom in on this subset of candidate substrates. And so here, what we can immediately appreciate is that for this particular substrate, DL48, we not only observe very strong selectivity for MMP13, but remarkable efficiency relative to the top performing efficient substrate from the training set that is shown here in blue, as well as a CleaveNet generation that was nominated for efficiency. So already this was quite encouraging, and it became even reinforced when now we look at things at the sequence level and are able to, again, pull out some distinctions from a composition point of view of sequence level determinants and amino acid patterns that seem to be driving the particular composition of these sequences that are highly efficient and highly selective MMP13 substrates. And so in hand, together, this gave us a very strong validation that we could actually use this approach to design substrates in a targeted way that were not only selective, but also efficient for a target protease of interest. So ultimately, this work has brought together not only two kind of distinct models from a modeling point of view in synergizing the complementary uses of a predictive model and a generative model, but also we're quite encouraged by this approach overall in terms of establishing a pipeline for guided substrate design for protease enzymes, which we were able to validate experimentally through this large-scale in vitro screen. Moving forward, we are quite interested in possibly taking some of these design substrates and moving them towards these clinical applications in conditionally activated diagnostics or therapeutics from an experimental point of view, but also, stepping back and thinking about how this work fits into the protein engineering and design community at large. While there's been a lot of, attention to the design of enzymes through either sequence approaches or structure approaches, the design of substrates for those enzymes has been less appreciated. And so we're hoping that this approach and this framework can provide a perspective for those who are potentially interested in substrate or inhibitor design applications for enzymes to think about how this work can be more generalizable towards different enzyme classes and use cases. So with that, I will conclude and thank especially the collaborators across MSR, Kevin and Sarah in particular, and as well as at MIT in Sangeeta Bhatia's lab. And I'll glad to have a little bit of time remaining for any questions that you all may have.